Hello and welcome to our presentation based on the post-translational modification of osteopontin. This experiment was carried out by scientists mentioned on this slide and will be commented on by me, Mariam, the group leader, and my colleagues Yasmin and Ayan. The aim of the experiment is to test the hypothesis that post-translational modification of the sibling family of proteins in general, and osteopontin in particular, can modify the abilities of these proteins to regulate the in vitro hydroxyapatite mineral crystallite formation and growth, depending on the level or extent of phosphorylation. This is a very important study, as scientific studies and research proves that OPN deficiency in mice leads to enhanced mineralization in bones, leading to vascular calcification. Vascular calcification is the presence of progressively enlarging deposits of calcium minerals in the major arteries. This is a common complication of chronic kidney disease and promotes congestive heart failure by eroding compliance and elastance of the arteries. Osteopontin is clinically proven to significantly reduce the accrual of vascular calcification. Osteopontin is a member of the sibling family proteins that is important for regulation of biomineralization. Phosphorylated bone osteopontin in cell-free in vitro systems inhibits HA mineral deposition. Our hypothesis is that post-translational modifications such as phosphorylation and fragmentation determines the ability of OPN to regulate hydroxyapatite formation. Osteopontin, also known as OPN, is a highly phosphorylated sialoprotein and a human gene product and a well-known sibling glycoprotein. It was first discovered in 1986 in osteoblasts. The prefix osteo alludes to the fact that the protein is expressed in bone, while the suffix pon is Latin for bridge, which means and signifies osteopontin's role as a linking protein. Hydroxyapatite, HA, is a naturally occurring mineral form of calcium apatite which can be found in teeth and bones. It is commonly used as a filler to replace amputated bone or as a coating to promote bone ingrowths. Sibling proteins refers to the family of non-collagenous proteins which are small integrin binding ligands and linked glycoproteins. Mineralization mechanisms refers to the process by which minerals, biological tissues, are incorporated into soft matrices to either form a protective shield or structural support. An example of such a mineral is hydroxyapatite mentioned earlier.
The following are the materials and methods. The first one is purification and thrombin cleavage of milk osteopontin. Osteopontin was purified from bovine milk for generation of N-terminals, central and C-terminals fragments. Purified milk osteopontin was incubated with bovine thrombin in 0.1 molar ammonium bicarbonate at 37 Celsius for 1 hour. The second method is despheralization of milk osteopontin. Osteopontin fragments were incubated with bovine alkaline phosphate in 10 millimolars ammonium bicarbonate at a pH of 8.5 overnight at 37 Celsius and subsequently analyzed by mass spectrometer. The third method is binding of milk osteopontin fragments to collagen. The milk osteopontin fragments association with the collagen fibril was determined using binding studies with 50 Ug per ml dried fibrillar collagen per microtheta well, incubated in the presence of 1 millimolar calcium chloride with increasing concentration between 0.05 to 50 Ug per ml. Milk osteopontin or the N-terminal fragments, followed by washing and detection of binding by anti-milk osteopontin, followed by detection with an ALP-coupled second antibody.
From the data collected, the molecular sizes of the fluorescent milk osteopontin and each of the milk osteopontin studied in this experiment are summarized in this table. This table shows the full-length milk OPN and the N-terminal, C-terminal and the central SKK fragments generated by the thrombin cleavage of the bovine milk OPN, which was subsequently separated by gel filtration. From the mass differences calculated, we can see that there is no evidence that fluorescent milk osteopontin was forming aggregate or higher molecular weight species. Similar molarities of the fragments based on this data had distinct effect on the HA formation in the gelatin gel. The curve fit data in this figure also shows that all the concentrations tested, the N and the C terminal fragments promoted mineralization, but the SKK in contrast was an inhibitor. Apart from this, the experiment yielded no statistically significant values. In this figure, all three fragments inhibited HA-seeded growth in the pH stat and the gelatin gel system in comparison to the uncoated HA. The central SKK fragment was the most effective inhibitor in the pH stat. In this figure, the central SKK terminal lost its inhibitory effect, but the N-terminal fragment and the C-terminal fragment acted as an inhibitor when coated on fibrillar collagen. As you can see from the rate of HA formation values, the N-terminal and the C-terminal had competitively higher rates of HA formation than the SKK, which was significantly lower.
In conclusion, this study confirms the hypothesis that proteolytic fragments of milk osteopotin generated by thrombin cleavage have distinct effect on hydroxyapatite formation and growth. There is a colligid binding domain in osteopotin from binding domain of 150 to 177, which is contained in the SKK fragment. The fragments studied were derived from milk osteopotin, which may be distinct from the less phosphorylated forms of osteopotin isolated from the mineralized tissues. Recently, however, osteopotin purified from murine osteoclasts was shown to contain 21 phosphate groups distributed over 27 sites, implying that the osteopotin first made in bone may be more highly phosphorylated than that isolated from the mineralized tissues. These are our references.
I hope you have enjoyed this presentation. Thank you for listening.